Cognitive neuroscience, the brain in action. Cognitive neuroscience involves intensive study of the brain as well as behaviour. Alas, the brain is complicated, to put it mildly. It consists of 100 billion neurons, and these neurons are connected in very complex ways. To understand research involving functional neuroimaging, we must consider how the brain is organised and how the different areas are described. Various ways of describing specific brain areas are used. Below we will discuss the three main ones. First, the cerebral cortex is divided into four main divisions or lobes. There are four lobes in each brain hemisphere: frontal, parietal, temporal and occipital. The frontal lobes are divided from the parietal lobes by the central sulcus. Sulcus means a furrow or groove, and the lateral fissure separates the temporal lobes from the parietal and frontal lobes. In addition, the parieto-occipital sulcus and pre-occipital notch divide the occipital lobes from the parietal and temporal lobes. The main gyri, which are ridges, or gyrus is the singular, within the cerebral cortex are shown in figure 1.3. Researchers use various terms to describe more precisely the area or areas of the brain activated during the performance of a given task. The key terms here are sulcus, which is the groove or the furrow in the surface of the brain, the gyri, which is the prominent elevated areas or ridges of the brain's surface. Gyrus is the singular, so the sulcus is the groove, the gyri is the raised bit. Dorsal means superior or towards the top of the brain. Ventral means inferior or towards the bottom of the brain. Rostral is anterior or towards the front of the brain, and posterior is towards the back of the brain. Lateral is situated at the side of the brain, and medial is situated in the middle of the brain. I sort of think of the sulcus and the gyri as mountains and valleys. So the sulcus would be the valley and the gyri would be the mountain. And I'm just going to go over this really quick. So dorsal, top; ventral, bottom; rostral, front; posterior, back; lateral, side; medial, middle. So there we just discussed the cerebral cortex and the four main divisions or lobes. Second, the German neurologist Brodmann produced a map of the brain based on differences in the distributions of cell types across cortical layers. So it might be an idea to look up Brodmann with a double N, brain map. Brodmann identified 52 different areas, and we will often refer to areas such as BA17, which simply means Brodmann Area 17. Third, we can focus on the functions of different brain areas. For example, Brodmann Area 17. This is often referred to as primary visual cortex because it is an area strongly associated with the early processing of visual stimuli. Brain network organisation. Bullmore and Sporns argued that two major principles might determine brain organisation. First, there is the principle of cost control. Costs, example in terms of energy and space, would be minimised if the brain consisted of limited short-distance connections. Second, there is the principle of efficiency. Efficiency in terms of the ability to integrate information across the brain. This can be achieved by having very numerous connections, many of which are long distance. The problem is that these two principles are in conflict. You cannot have high efficiency at low cost. You might think it would be best if our brains were organised purely on the basis of efficiency. However, this would be hugely costly. If all 100 billion neurons in the brain were interconnected, the brain would need to be 12.5 miles wide. In fact, neurons mostly connect with nearby neurons, and no neuron is connected to more than about 10,000 neurons. As a result, the human brain has achieved a near optimal trade-off between cost and efficiency. Our brains are reasonably efficient, and this has been achieved at manageable cost. Within our brain network, there are modules, small areas of tightly clustered connections, and hubs, regions that have large numbers of connections to other regions. These hubs include areas, example the anterior cingulate, associated with high-level cognitive processes and consciousness. What light does a focus on brain network organisation shed on individual differences in cognitive ability? Van der Heuvel and others found IQ did not correlate with the total number of brain network connections. However, there were impressive associations between IQ and the global efficiency of functional brain networks. The correlation was plus 0.75 in medial prefrontal cortex, BA9/10, and plus 0.72 in inferior parietal regions, BA39/40. Thus, how the brain is wired has important implications for the efficiency of cognition. Techniques for studying the brain. Introduction. Technological advances mean we have numerous exciting ways of obtaining detailed information about the brain's functioning and structure. In principle, we can work out where and when specific cognitive processes occur in the brain. This allows us to determine the order in which different brain areas become active when someone performs a task. It also allows us to find out whether two tasks involve the same brain areas in the same way or whether there are important differences. Major techniques used to study the brain are, and I'll do these in their abbreviations first and then I will elaborate on them. So the major techniques are single unit recording, ERPs, PET, fMRI, EFMRI, MEG, TMS, and RTMS. So starting with the first one, single unit recording. This technique, also known as single cell recording, involves inserting a microelectrode 1/10,000th of a millimetre in diameter into the brain to study activity in single neurons. It is very sensitive since electrical charges of as little as 1/1,000,000th of a volt can be detected. ERPs, which are event-related potentials. The same stimulus, or very similar ones, is presented repeatedly, and the pattern of electrical brain activity recorded by several scalp electrodes is averaged to produce a single waveform. This technique allows us to work out the timing of various cognitive processes very precisely, but its spatial resolution is poor. PET, which is positron emission tomography. This technique involves the detection of positrons. Positrons are atomic particles emitted from some radioactive substances. PET has reasonable spatial resolution but poor temporal resolution and measures neural activity only indirectly. fMRI, which is functional magnetic resonance imaging. This technique involves imaging blood oxygenation using an MRI machine. Functional MRI has superior spatial and temporal resolution to PET, but also provides an indirect measure of neural activity. EFMRI, event-related functional magnetic resonance imaging. This involves separating the elements of an experiment into discrete points in time so that the cognitive processes and associated brain responses associated with each element can be analysed independently. Event-related fMRI is generally very informative and has become markedly more popular recently. MEG, magnetoencephalography. This technique involves measuring the magnetic fields produced by electrical brain activity. It provides fairly detailed information at the millisecond level about the time course of cognitive processes, and its spatial resolution is reasonably good. TMS, transcranial magnetic stimulation. This is a technique in which a coil is placed close to the participant's head and a very brief pulse of current is run through it. This produces a short-lived magnetic field that is generally, but not always, inhibits processing in the brain area affected. When the pulse is repeated several times in rapid succession, we have repetitive transcranial magnetic stimulation, RTMS. Repetitive TMS is used very widely. It has often been argued that TMS or RTMS causes a very brief lesion, a lesion being a structural alteration caused by brain damage. This technique has, jokingly, been compared to hitting someone's brain with a hammer. The effects of TMS are often more complex than suggested so far. In fact, there is often interference because the brain area to which TMS is applied is involved in task processing as well as the activity resulting from the TMS stimulation. And all of those are key terms, so I have no idea how we're supposed to remember those, so I'll go over them again from the key terms box on the side. Single unit recording is an invasive technique for studying brain function, permitting the study of activity in single neurons. Event-related potentials, or ERPs, which is the pattern of electroencephalograph, EEG, activity obtained by averaging the brain responses to the same stimulus or very similar stimuli presented repeatedly. Positron emission tomography, or PET, is a brain scanning technique based on the detection of positrons. It has reasonable spatial resolution but poor temporal resolution. Functional magnetic resonance imaging, fMRI, a technique based on imaging blood oxygenation using an MRI machine. It provides information about the location and time course of brain processes. Event-related functional magnetic resonance imaging, EfMRI. This is a form of functional magnetic resonance imaging in which patterns of brain activity associated with specific events, example, correct versus incorrect responses on a memory test, are compared. And magnetoencephalography, MEG, a non-invasive brain scanning technique based on recording the magnetic fields generated by brain activity. Which technique is best? There is no single or simple answer. Each technique has its own strengths and limitations, and so experimenters match the technique to the research question. At the most basic level, the various techniques vary in the precision in which they identify the brain areas active when a task is performed, spatial resolution, and the time course of such activation, temporal resolution. Thus, the techniques differ in their ability to provide precise information concerning where and when brain activity occurs. High spatial and temporal resolutions are advantageous if a very detailed account of brain functioning is required. In contrast, low temporal resolution can be more useful if a general overview of brain activity during an entire task is needed. What a mouthful. I just want to add, I only have a black and white printer here, and it's the worst printer, and the key terms are in a coloured box, so that comes out grey with black writing on it, so it's very difficult for me to see it, I'm afraid. Techniques for studying the brain, detailed analysis. We have introduced the main techniques for studying the brain. In what follows, we consider each one in more detail. Oh God, it goes over all those techniques again. Single unit recording. The single unit or cell recording technique is more fine-grained than any other technique. However, it is invasive and so rarely used with humans. An interesting exception is a study by Quiroga and others on epileptic patients implanted with electrodes to identify the focus of seizure onset. A neuron in the medial temporal lobe responded strongly to pictures of Jennifer Aniston, but not to pictures of other famous people. Weird. This finding needs to be interpreted carefully. It is highly improbable that only a single neuron responds to Jennifer Aniston. Only a tiny fraction of the neurons in the medial temporal lobe was studied. Event-related potentials. EEG, electroencephalography is based on recordings of electrical brain activity measured at the surface of the scalp. Very small changes in electrical activity within the brain are picked up by scalp electrodes and can be seen on a computer screen. However, spontaneous or background brain activity can obscure the impact of stimulus processing on the EEG recording. The answer to the above problem is to present the same stimulus or very similar stimuli several times. After that, the segment of EEG following each stimulus is extracted and lined up with respect to the time of the stimulus onset. These EEG segments are then averaged together to produce a single waveform. This method produces event-related potentials, or ERPs, from EEG recordings and allows us to distinguish genuine effects of stimulation from background brain activity. I have no idea what I just read. Actually, I have no idea what I've read for the past 15 minutes. It's all just one big blur of abbreviations and words ending in ography. ERPs have excellent temporal resolution. How about that? Indeed, they can often indicate when a given process occurred to within a few milliseconds. The ERP waveform consists of a series of positive, P, and negative, N, peaks, each described with reference to the time in milliseconds after stimulus presentation. For example, N400 is a negative wave peaking at about 400 milliseconds. ERPs provide very detailed information about the time course of brain activity. A behavioural measure, for example, reaction time, typically provides only a single measure of time on each trial, whereas ERPs provide a continuous measure. However, ERPs do not indicate with precision which brain regions are most involved in processing, in part because skull and brain tissues distort the brain's electrical fields. In addition, ERPs are mainly of value when stimuli are simple and the task involves basic processes. Example, target detection, triggered by task stimuli. Finally, we cannot study most complex forms of cognition, example, problem solving with ERPs. Oh, I missed a key term, one of the techniques, which was transcranial magnetic stimulation, or TMS, which is a technique in which magnetic pulses briefly disrupt the functioning of a given brain area. It is often claimed that it creates a short-lived lesion. More accurately, TMS causes interference when the brain area to which it is applied is involved in task processing as well as activity produced by the applied stimulation. There was also the RTMS, repetitive transcranial magnetic stimulation, which is the administration of transcranial magnetic stimulation several times in rapid succession. Lesions. Lesions are structural alterations within the brain caused by disease or injury. And EEG, I'm not even going to say it this time, EEG, recording the brain's electrical potentials through a series of scalp electrodes. Okay, back to the detailed analysis. Positron emission tomography, PET, is based on the detection of positrons, atomic particles emitted by some radioactive substances. Radioactively labelled water, the tracer, is injected into the body and rapidly gathers in the brain's blood vessels. When part of the cortex becomes active, the labelled water moves there rapidly. A scanning device measures the positrons emitted from the radioactive water, which leads to pictures of the activity levels in different brain regions. Note that tiny amounts of radioactivity are involved. PET has reasonable spatial resolution in that any active brain area can be located to within 5 to 10 millimetres. However, it has very poor temporal resolution. PET scans indicate the amount of activity in any given brain region over 30 to 60 seconds. Magnetic resonance imaging, MRI and fMRI. So MRI involves the use of an MRI scanner, look that image up, containing a very large magnet weighing up to 11 tonnes. A strong magnetic field causes an alignment of protons, subatomic particles, in the brain. A brief radio frequency pulse is applied, which causes the aligned protons to spin and then regain their original orientations, giving up a small amount of energy as they do so. The brightest regions in the MRI scan are those emitting the greatest energy. MRI scans can be attained from numerous angles, but tells us only about brain structure rather than its functions. Happily enough, the same principles used to produce MRI can also be used to provide functional information in the form of functional magnetic resonance imaging, fMRI. Oxyhemoglobin is converted to deoxyhemoglobin when neurons consume oxygen, and deoxyhemoglobin produces distortions in the local magnetic field. This distortion is asserted by fMRI and provides a measure of the concentration of deoxyhemoglobin in the blood. Technically, what is measured in fMRI is known as BOLD, B-O-L-D. So this is a key term. It's blood oxygen level dependent contrast. Blood oxygen level dependent contrast. This is the signal measured by fMRI. Changes in the BOLD signal produced by increased neural activity take some time to occur, so the temporal resolution of fMRI is about 2 or 3 seconds. However, its spatial resolution is very good, approximately 1 millimetre. Since fMRI has superior temporal and spatial resolution to PET, it has largely replaced it in neuroimaging research. Suppose we want to understand why our participants remember some items but not others. We can use event-related fMRI, in which we consider each participant's patterns of brain activation for remembered and forgotten items. Wagner and others recorded fMRI while participants learned a list of words. There was more brain activity during learning for words subsequently recognised than those subsequently forgotten. These findings suggest forgotten words were processed less thoroughly than remembered words at the time of learning. fMRI's limitations. First, it provides an indirect measure of underlying neural activity. Second, there are distortions in the bold signal in some brain regions, example close to sinuses, close to the oral cavity. Third, the scanner is noisy, which can cause problems for studies involving auditory stimuli. Fourth, some people, especially those with claustrophobia, find it uncomfortable to be encased in the scanner. Cook and others found 43% of participants in an fMRI study were at least a bit upset by the experience, and 33% reported side effects, example headaches. Fifth, there are constraints on the kinds of stimuli that can be presented to participants lying in a scanner. There are also constraints on the response they can be asked to produce, because even small movements can distort the bold signal. And remember, bold was blood oxygen level dependent contrast. So I'm not sure why it's not bold C. I saw a quote or something once that said, "Never mock a person who mispronounces a word. It means they read." So please keep that in mind with all my mispronunciations, like the next one, which is MEG, magnetoencephalography. So I'm not exactly sure how to pronounce that. So MEG involves using a, get ready for it, superconducting quantum interference device, SQUID, to measure the magnetic fields produced by electrical brain activity. The technology is complex, like the words, because the size of the magnetic field created by the brain is extremely small relative to the Earth's magnetic field. Wow, now we're getting deep and further away from psychology. However, MEG provides very accurate measurement of brain activity, in part because the skull is virtually transparent to magnetic fields. Bet you didn't know that. MEG has excellent temporal resolution at the millisecond level and often has very good spatial resolution as well. However, it is extremely expensive. In addition, some people find it uncomfortable to take part in MEG studies. Cook discovered that 35% of participants found the experience a bit upsetting, and the same percentage reported side effects such as muscle aches or headaches. And the last one in that list is the TMS, transcranial magnetic stimulation. TMS is a technique in which a coil, often in the shape of a figure of 8, is placed close to the participant's head. A very brief, less than 1 millisecond, but large magnetic pulse of current is run through it. This causes a short-lived magnetic field that generally, but not always, leads to inhibited processing in the affected area, generally about 1 cubic centimetre in extent. More specifically, the magnetic field created leads to electrical stimulation in the brain. In practice, several magnetic pulses are typically given in a short period of time. This is repetitive transcranial magnetic stimulation, RTMS. Most research has used RTMS, but we will often simply use the more general term TMS. And the picture looks like, it just looks like some sort of bowl on the person's head with a pair of clear glasses, like you wear at the dentist or getting laser, and a tube thing connected. It doesn't look like a figure 8, this picture that they're showing. So it might be an idea to look it up. Not that I really think it matters. Anyway, back on tangent. What is an appropriate control condition against which to compare the effects of TMS or RTMS? One possibility is to compare task performance with and without it. However, TMS creates a loud noise and muscle twitching at the side of the forehead, and these effects might lead to impaired performance. Applying TMS to a non-critical brain area, irrelevant to task performance, is often a satisfactory control condition. The prediction is that task performance will be worse when TMS is applied to a critical area than a non-critical one. Why are TMS and RTMS useful? As mentioned earlier, they often create a temporary lesion, so the role of any given brain area in task performance can be assessed. If TMS applied to a particular brain area impairs task performance, we can conclude that brain area is necessary for task performance. Conversely, if TMS has no effects on task performance, then the brain area affected by it is not necessary. Thus, we can often make stronger causal statements about the brain area's underlying performance with TMS than with most other techniques. TMS can also indicate when any given brain area is most involved in task performance. For example, Krakow and others asked participants to detect letters. Performance was maximally impaired when TMS was applied to occipital cortex 80 to 100 milliseconds after letter presentation rather than at shorter or longer delays. Evaluation. In principle, the greatest advantage of TMS and RTMS over neuroimaging techniques is that they increase our confidence that a given brain area is necessary for task performance. TMS allows us to manipulate the availability of a brain region for involvement in the performance of some cognitive task. In contrast, we only establish associations or correlations between activation in various brain areas and task performance with functional neuroimaging. TMS can be regarded as producing a brief lesion. However, it has various advantages over research on brain-damaged patients having genuine lesions. For example, we can compare any given individual's performance with and without a lesion with TMS, but this is rarely possible with brain-damaged patients. In addition, the experimenter controls the brain areas affected by TMS, but such control is impossible with brain-damaged patients. What are TMS's limitations? First, its effects are complex and not fully understood. For example, Allen and others found RTMS applied to early visual cortex of cats not engaged in any task caused an increase of spontaneous brain activity lasting up to 1 minute. However, activity in the visual cortex produced by viewing gratings was reduced by up to 60% by RTMS. The effects of TMS on performance are generally negative but sometimes positive. Why does TMS sometimes enhance performance? Consider an area X that typically inhibits the functioning of area Y. TMS applied to area X will reduce this inhibition and so might enhance the functioning of area Y. More generally, brain functioning is remarkably complex, and so we would expect TMS to have somewhat variable effects. Second, it has proved difficult to establish the precise brain areas affected by TMS, some of which can be distant from the stimulation point. However, progress can be made by combining TMS with neuroimaging techniques to clarify the effects of TMS on brain activity. Third, TMS can only be applied to brain areas lying underneath the skull, but not to areas with overlying muscle. That limits its overall usefulness. And fourth, there are safety issues with TMS. For example, it has very occasionally caused seizures in participants in spite of stringent rules designed to ensure their safety. The overall strengths. Do the various techniques for studying the brain answer all of our prayers? That would be an overstatement. However, cognitive neuroscientists have made substantial contributions to our understanding of human cognition. The major strengths of cognitive neurosciences will be discussed here. First, cognitive neuroscience has increasingly helped to resolve theoretical controversies that had remained intractable with purely behavioural studies. Here are 2 examples. The first concerns speech perception. Listeners presented with degraded speech find it more intelligible when the words are predictable. The crucial issue is when knowledge of what is being presented influences speech perception. It might occur early and thus directly affect basic auditory processes. Alternatively, it might occur late and only after basic auditory processing has finished. Theorists differ in their favoured explanation. Wild and others address this issue. Listeners heard degraded speech accompanied by visual stimuli that matched or did not match the auditory input. There was more activity in primary auditory cortex involved in early auditory processing when the visual input matched the auditory input than when it did not. This strongly suggests knowledge of what was being presented directly affected basic auditory processes. The second example concerns visual imagery. There has been much controversy as to whether visual imagery resembles visual perception. Most behavioural evidence is inconclusive. However, brain imaging research has shown 2/3 of the brain areas activated during visual perception are also activated during visual imagery. Coslin and Thompson found in a meta-analysis that even brain areas involved in early stages of visual perception are often activated during visual imagery. The above findings strongly suggest the processes in visual imagery resemble those in visual perception. However, R.J. Lee and others identified important differences using neuroimaging. Participants viewed or imagined common objects, and attempts were made to decide which objects were involved based on patterns of brain activation. Identification of perceived objects was much better based on activation in early visual processing areas than subsequent ones, whereas the opposite was the case for imagined objects. Thus, there is more involvement of low-level visual processes in the perception than imagery. Second, it is a major challenge to understand the complexities of the cognitive system and its underlying brain organisation. As we saw earlier, Bullmore and Sporns argued that the brain is organised into many tight clusters or modules, plus long-distance connections among them. The incredible richness of the data obtained from neuroimaging studies mean cognitive neuroscientists can, at least in principle, construct theoretical models accurately mimicking the complexities of brain functioning. In contrast, cognitive neuropsychology seems less flexible and more committed to the notion of a modular brain organisation. Third, another advantage arises from the remarkable richness of neuroimaging data. If it becomes clear that one approach to analysing such data is limited, it is easy to reanalyse them within a different theoretical framework. For example, it used to be assumed that most face processing occurs in the fusiform face area, but this was a substantial oversimplification. An approach based on the assumption that face processing involves a network of brain regions provides a more accurate account. Thus, cognitive neuroscience can be self-correcting. More generally, cognitive neuroscientists attach less importance than they used to the assumption of functional specialisation, the notion that each brain region is specialised for a different function. Instead, they accept there is substantial integration and coordination across the brain. Such functional integration can be studied by correlating activity across different brain regions. If a network of brain areas is involved in a given process, activity in all of them should be positively correlated when the process occurs. There is strong evidence for such functional integration with conscious perception, which seems to depend on coordinated activity across several brain regions. Cognitive neuroscientists have identified an increasing number of major brain networks. For example, Corbetta and Schulman used findings from cognitive neuroscience to identify 2 attention networks, one concerned with goal-directed attention and the other with stimulus-driven attention. Fourth, cognitive neuroscience is often especially useful when combined with other approaches. Here is an example based on the notion that recognition memory depends on 2 different processes, recollection and familiarity. These processes differ in that only recollection involves the conscious retrieval of contextual information. It has been argued theoretically that recollection involves the hippocampal system, whereas familiarity involves the nearby peripheral system. Research within cognitive neuropsychology has produced a double dissociation supporting the above theory. Patients with damage to the hippocampus have impaired recollection but intact familiarity, whereas those with damage to the peririnal, PERIRHINAL system have impaired familiarity but intact recollection. Neuroimaging research has strengthened the support for the theory. Recollection is associated with more activation in the hippocampus than the peririnal cortex, whereas the opposite is the case for familiarity. And then general limitations. We turn now to various issues raised by cognitive neuroscience. First, cognitive neuroscientists often overinterpret their findings by assuming there are one-to-one links between cognitive processes and brain areas. Thus, for example, activation in a small brain region, in brackets a blob, is interpreted as being the love area or the religion area. This approach has been referred to unflatteringly as blobology. There's a key term here, reverse inference. As applied to functional neuroimaging, it involves arguing backwards from a pattern of brain activation to the presence of a given cognitive process. Blobology is in decline. However, there is still undue reliance on reverse inference. A researcher infers the involvement of a given cognition process based on an activation within a given brain region. Here is an example. Individuals exposed to threat-related information typically show activation of the amygdala, part of the limbic system. This led many researchers to conclude the amygdala is central to a fear system. What is wrong with the above conclusion? Other researchers have shown the processing of most emotions is associated with amygdala activation. This illustrates a key problem with reverse inference. Most brain regions are involved in several different cognitive processes, and so activation of any given brain area is not very informative. This was shown clearly by Yarkoni and others, who considered areas of brain activation across 3,489 studies. Some brain areas, example, the dorsolateral prefrontal cortex, anterior cingulate cortex, and anterior insula, were activated in 20% of the studies. Such areas are involved in several different cognitive processes. Second, it is very difficult to bridge the divide between psychological processes and concepts on the other hand, and patterns of brain activation on the other. As Harley pointed out, we may never find brain patterns corresponding closely to psychological processes such as attention or planning. Harley concluded as follows: our language and thought may not divide up in the way in which the brain implements these processes. Third, most neuroimaging studies are underpowered, typically using 20 participants or fewer. This produces the problem that most fMRI analysis will detect only a fraction of the true effects, producing a deceptive illusion of selective activation. One solution to this problem is to combine findings across studies. Fourth, false positive results, i.e. mistakenly concluding that random activity in a given brain area is task-relevant activation, are common and may occur as often as 15% of the time. False positives arise because most neuroimaging studies produce huge amounts of data, and some researchers fail to correct their statistical thresholds, p-values required for significance, to take full account of it. Bennett and others provided an amusing example of a false positive finding. They asked their participant to determine the emotions drawn in pictures of people in social situations. When they did not correct their statistical thresholds, there was significant evidence of brain activation. The interesting feature of this study was that the participant was a dead salmon. Thus, we know for certain the finding was a false positive. Fifth, most brain imaging techniques reveal only associations between patterns of brain activation and behaviour. For example, performance on a reasoning task is associated with activation of the prefrontal cortex. Such associations are purely correlational and do not show that the brain regions activated are essential for task performance. Brain activation might also be caused by participants engaging in unnecessary monitoring of their performance or attending a non-task stimuli. TMS offers a partial solution to this causality issue. We can show a given brain area as necessary for the performance of a task by finding that TMS disrupts that performance. However, the complexities of TMS's effects on the brain sometimes mean that the caution in interpretation is needed. 6. Many cognitive neuroscientists assume that most brain activity is driven by environmental or task demands. If that assumption is correct, we might expect most brain activity in response to such demands. Surprisingly, that is not the case. In fact, the increased brain activity occurring when someone performs a cognitive task typically adds less than 5% to resting brain activity. This is probably much less than the brain energy consumption devoted to intrinsic activity within the brain that occurs in unstimulating environments. Why is the brain so active even when the environment is unstimulating? Part of the answer is that people often devote cognitive resources to predicting future environmental changes. However, the finding that patterns of brain activity are smaller in different states of consciousness, including coma, anesthesia, and slow wave sleep, suggest most intrinsic brain activity reflects basic brain functioning. As a result of intrinsic brain activity, task performance is often associated with decreased brain activity in some brain regions rather than the expected increase. Seventh, cognitive neuroscience shares with cognitive psychology problems of ecological validity, applicable to everyday life, and paradigm specificity. Findings do not generalise across paradigms. Indeed, the problem of ecological validity may be greater in cognitive neuroscience. Participants in fMRI studies, the most used technique, lie on their backs in somewhat claustrophobic and noisy conditions and have very restricted movement, not much like everyday life. Kutchis and Park compared the effects of being an MRI scanner or simply in the laboratory on long-term recognition memory. Memory was significantly worse in the scanner, presumably because it provided a more distracting or anxiety-creating environment. Eighth, we must avoid the neuroimaging illusion, which involves overvaluing the contribution pictures of brain activity make to our understanding of human cognition. Keener and others presented neuroscience articles accompanied by brain images. The more three-dimensional the images appeared to be, the more positively the articles were evaluated. Here is a concrete example of the neuroimaging illusion. On 28th of August 2007, The Guardian had the following headline: "Brain scans pinpoint how chocoholics are hooked." In essence, the researcher involved had found that the sight of chocolate produced more activation in reward centres of the brain in chocolate cravers than in non-cravers. Thus, the findings that so impressed The Guardian tell us little more than that chocolate cravers find chocolate rewarding.